Hello. Hello guys, am I audible? Okay, thank you. Uh, so I think it's 7:05 and we can start now. Okay, so I'll be sharing my screen. So are you guys able to see my screen? Okay, yeah. Okay. So okay. So I hope am I audible and my screen is visible to you all. And if you are facing some problems, then please do let me know by unmuting yourself and speak that if something problem you are facing. So yeah, welcome you all to the week two session of the doubt and problem solving for the course of techniques of materials characterization. And I hope the last session that where we solved numerous problem related to the week one lectures. I hope you guys were able to solve the problems and were successfully did the assignment. So I'm hoping the same motivation for this week two also, that today we'll be sharing or we will be try to discuss some concepts and related to problem and related some concepts, and hopefully that will not be exactly similar, but yeah, I can say that that will be really gonna help to solve the assignment problem, as well as that will clear your concept for the week second lecture. So let's start for the today's session, and yeah, again, whoever is joining for the first time, so again, I'll give my introduction. So my name is Saurabh Soni, and currently I'm a PhD research scholar at IIT Bombay, and here I'm a, Prime Minister Research Fellow, and I will be the additional TA for this course, where I will be solving your doubts and the problem related to the assignment that will help to learn better and more efficiently. So let's start for the today's session. And before that, let me check if anyone's in there in the yeah, no one is there. So I hope you guys are able to see my screen. So okay, let's begin with our first question. So I hope you had some you have gone through the lectures before this doubt session. So the first question that I'll be going to take is the first question that I will be going to take is, in which type of optical microscope both the light source and the objective lens are present in the same side of the specimen? So your options that you have gone through the microscopy, that in the last lectures we have covered that what is microscopy. So microscopy is nothing but the objects. There are many things in the field of in all field in the field of science and technology which we cannot see by our naked eye. So we need a microscopy. See in so in the microscopy generation, there are different kind of microscopy. Scientists are keep on evolving a different microscopy for having a better understanding of the object, and try to learn, because whatever, if we can see with a much better magnification and the better resolution, we will be able to understand the more exactly what is phenomena is occurring inside the material. So that will help. So here it is asking about a very basic question. So it is about the some setup that where the light source and the objective lens are present in the same side of a specimen. So maybe you all have gone through the lectures, and the options are reflected light microscopy, transmitted light microscopy, and the both, and none of this. So can anybody guess if you have gone through the lectures? So I would ask to you guys. So please guess, then we will be discussing about this. So please guess or answer if you know. Option A. Okay. Thank you. So, so guys, the main motivation behind this uh, asking to you is to keep awaking to you guys, so that you can also participate in this, and otherwise I can just go through the questions and I can discuss the answer and the concept related to this, but I don't want this. I want your participants also. So that's why I'm asking to you. So please answer if you know, and even if you don't know also, then also it's fine. So yeah, someone said okay, option B. Okay, fine. So let's check. Uh, okay. So this is the reflected light microscopy. So firstly, here we will see that okay. So yeah, firstly we will see this is the light source, and this is the collector lens, and this is your filament image at condenser aperture diaphragm. So here this is asking about the light source and the objective lens are present in the same side of the specimen. So here this is the light source, and where is the objective lens? Okay, so here this is your collector lens. Okay, so here you can see somewhere here condenser and the objective. Okay, and this is your image plane. So the light is coming, and also there are some aperture, and this is going through this beam splitter or the mirror, and through this specimen it is reflected, and then it is going to the image plane. So here in the reflected microscopy, the optical pathway for the reflected light begins with the illuminating rays originated in the lamp housing for the reflected light. So here the main fundamental is the reflection. Okay, here the light is not transmitting. So here the light source is this side and the objective lens is this side. Okay, and we will see the difference what is exactly in the transmitted light microscopy. Okay, so here this is the transmitted light microscopy. So here we have the source illumination, and here we have the condenser aperture, and this is our specimen, and here we are having objective rear focal plane. So where can we so this is our specimen, right, and this is our source. And so here you can see the specimen is at the between, it is not, sorry, yeah. So here you can see the objective is this side, and this is sorry, the condenser aperture is this side. Source, it is asking about the source and the objective. So source is this side, and in between there is a specimen, and the objective is the other side. So it is asking about the same. So here the light source and the objective is at the same side, but here it is not at the same side. So our answer would be the reflected light microscopy. Okay, so here also in this the main concept is the reflection, because here there is a beam splitter or the mirror, but here the light source, the light is coming, and in the center there is a specimen or the object, okay, which we want to see. So the light is transmitting through this specimen. Okay, so This method is used to distinguish the morphological characteristics and the optical properties of the observed material. Here, you can see here the also the condenser is able to control the angle. Okay, this is going at certain angle. So this is your condenser. Here this is a straight line, but here it is having some angle. So again, in your previous lecture you have seen that it is controlling the angle of the illumination, which permits the right balance of the resolution and the contrast in the microscopy. So here the light source and the objective is at the same side, so the answer would be the reflected light microscopy. So one who said the reflected, I think uh, he or she is correct. So the second is the cut. Okay, the second is the contrast generation in optical microscopy is increased by. So here it is asking about the optical microscopy. How can we increase the contrast generation? Okay, so I will also be discussing about the contrast, and I will show you some example that what is exactly the contrast is very important in the field of this microscopy. So the first one is the background subtraction algorithm. Second one, using stain or dye in biological specimen. Third one is both by background subtraction algorithm and using stain dye in biological specimen and mounting of specimen. So if anybody knows, then please answer, and if you know the some reasoning behind that, then also please say that why. Option. Okay, okay. So, okay, fine. So, do you have any reasoning behind that why the contrast can be, increased by using this background subtraction and this by using a dye? Or others can also answer if they knows. It's fine uh, if you do not have. Huh? Okay, okay, okay. Does anybody also having their opinion or something to say about this? Okay, fine. So let's check. So this you can find in the this slide. So firstly, the contrast. So maybe you all have gone through some images where you can say, okay, this is having a better contrast, this is having a poor contrast. So the contrast is what? So the contrast is given by this formula where this is the intensity of the specimen minus the intensity of the background into 100. Yeah. So the contrast is given by the intensity of the specimen, and this is the background intensity divided by the background intensity. So here the background intensity is having a very huge role to enhancing the contrast. Okay, so here if you deal with the background intensity, then you will have a better contrast. So here we can see the y-axis is the contrast. Okay, so we want to increase the contrast, and it is the image intensity, and here we are having a different of background intensity. Okay, so for an example here, this is okay, so this is if you will have a background intensity of 0.5. Okay, so for the same image intensity, the contrast is the, suppose if you will take a point to image intensity. Okay, so the contrast is less, but when we are going from zero point 0.5 to zero point 0.1 to zero point 0.01, we are decreasing the intensity of the background. Then for the same image intensity, the contrast is improving or the contrast is increasing. Okay, so we can say that by reducing or by decreasing the background intensity, we are able to increase the contrast. Okay, so this is one thing. So here also we can see when the background is bright, the human eye requires local intensity fluctuation of at least 10 to 20% to be able to recognize this. Okay, so suppose if I will give you some two similar kind of picture, and that is having a contrast or the contrast is less than that 10%, then it might be possible that your eye would not be able to see the clear difference of the both the image. But I will change a contrast much enough more than the 20 or 25%, then your eyes can be able to recognize, okay, this is having a less contrast, this is having a better contrast. So this is saying about this. So here we got one answer, which is the this. So also you can see the background intensity. So how, so this is the bright field illumination. Maybe in the lectures you have gone, but I will show you by this image. So this is the bright field image, this is the dark field image. Okay, so here this is the background intensity is high. Okay, so here because this is a bright field, and here the background intensity is low because this is the black color. So you are clearly able to see that which one you can see a better contrast image, or which one you can see that this is more better view. So obviously where the background intensity is less, which is looking better. So here there are many images. When you search in any research paper, or you can just Google search on this, that about the contrast, you will get a lots of image where you can easily see that when the background intensity is less, then your image will be, the contrast of your image will be much more clearer. Okay, so also the stain and dye. Okay, the someone said about the third, so which is the right answer, so which I will be discussed later also. So actually what is happening in the biological specimen or in the samples, that is very difficult to see. So here also, because this is a kind of light can transmit. So here also when we want to see the clear image, so when we use some kind of a dye or stain, then that will give you a better contrast. This is especially applicable for the biological samples. Okay, so for the other samples, suppose if you are having some metallic samples or some other samples, then you can reduce the background intensity. But instead of that, if you are going for the biological samples and your cells or the tissue is allowing to apply some kind of a stain or dye, then that would give a better contrast image. So yeah, the option is C. So this is again the question about the contrast enhancement. So is more sensitive to which mode? So there is a reflection mode, transmission mode, and both reflected and the transmission mode. So can anybody answer this question and why? Hello, those who are not speaking, please speak. Okay. So does anybody answer having some different answer? Somebody has said that transmission mode. Okay, so let's check. Yeah, so transmission mode is correct. Okay, so the Kanhan, so it is also very same guys. Why you are taking so much time? So as I, this is already discussed here. So you can see here. Okay, so in the transmitted, okay, so yeah, so in the transmitted mode, the your this light is, I mean, transmitted and then reflected, that is giving a bright illumination. Okay, and this is giving a, sorry, yeah. So in transmitted light microscopy, so here this is giving a, dark field. So that's why this will be the more sensitive to the contrast, because here your light is passing through the specimen, but in the reflected it is the contrast will be less. So here, because in the bright field the contrast comes from the absorbance of the light in the sample. Okay, but here the contrast comes from the light scattered by the sample. So here the answer will be the transmission mode. So, okay. So here the optical tricks are only used to amplify contrast difference in which microscopy. So in the second week lectures, you are having a different microscopy lecture. So like polarized microscopy, dark and bright field, phase contrast, and the DIC microscopy. So here it is asking about the optical tricks. So can anybody answer that which is used to amplify the contrast difference? So maybe if you know the setup or the arrangement of any of this microscopy, then you would be able to answer correctly. Can anybody say? There are many peoples, so I will also then ask you the name. Please, guys, say. Okay, I think then I will discuss. Okay, it's fine. So, okay, so can you see and guess that this arrangement, so that it would be easy for you to guess? Okay, so here this is this is asking about the amplify the contrast difference optical arrangement. Okay, so here this is the answer will be your phase contrast microscopy. So this is the setup of the phase contrast microscopy. So here it is asking about the phase contrast techniques uses optical tricks to translate phase shift into grayscale amplitude values. So here you have to go through the how the contrast is amplifying. So here in the phase contrast microscopy, there will be the phase change. Okay, we are using we because we will also having some slides where we will discuss in the detail. Okay, so where we will be discuss in the detail that so this is your source light, and there will be the condenser lens, and this is the specimen plane, and this is your objective, and here this will be your image plane. Okay, so in the phase contrast microscopy there will be the change in the phase, and that will be uh, because change in the phase, why there is a if there will be the change in the phase, this is because of the difference in the refractive index. Okay, so I will also having some further slides where I will be discussing that if there will be the difference in the refractive index, then there will be the change in the phase, and if there will be the change in the phase, the contrast will be developed. Okay, so here the waveform that have all been retarded to varying degrees by details in the specimen are superimposed in the intermediate image plane, where they amplify or attenuate each other, forming the final phase contrast image. Okay, so Yeah, so here you can also see there will be some constructive and a destructive interference will also occur. So this interference process in the intermediate image create bright and the dark areas of the various structures having refractive index variation in the specimen. So if there will be the refractive index variation, then there will be the phase difference will be generated, and that will be lead to give a better contrast. So this is the main thing. So if you will be going if you will be check in the lecture, and if you will find some difficulty, then please let me know. I will discuss detail about the working mechanism. But since the question was here, it is simply asking the contrast difference amplification, so where we are using the optical tricks or the arrangement, so the answer will be your phase contrast microscopy. So, okay, so which of this provides better contrast? So this I have already discussed. So it is very, very easy. Dark field microscopy, bright field microscopy, and so I want you guys please quickly answer this that I have discussed two, three times this. Please, anybody can answer. Bright field. Are you sure? It is asking about the better contrast. Okay, I will give you some hint. Now, this is a dark field, this is a bright field. Now, can you say the one who said the bright field? Can you again guess that which answer would be? Or you want to change your answer? Yes, yes, absolutely correct. It is also the same reasoning behind this question also. In the dark field microscopy, the background intensity will be the less. So, as per the formula of the contrast, the specimen sorry, the intensity of the specimen minus the intensity of the background upon intensity of the background. So if the intensity of the background will be the less, so which is very obvious in the here the intensity of the background is less, so the contrast will be the higher. So the answer will be your dark field microscopy. Okay. So again, as I said that phase contrast microscopy is popular for what? Opaque specimen, thin, transparent and stained biological samples, thin, transparent and unstained biological samples, all types of biological samples. So can anybody having some view or any opinion behind this that why we are using, because here our main purpose is not only to understand the various kinds of microscopy, but here the main thing as a scientist or as an engineer, we want to have some knowledge that okay, if we are having this kind of sample, then which kind of microscopy would be the better, because the application is very important. It's not just okay, we are having this kind of this sample, let's try by this microscopy. If it is not working, then let's go with this microscopy. So you should have some better clear you know uh, uh, idea in the mind. Okay, we are having this kind of specimen, then so which kind of microscopy we can use to have a better image. So whenever you are learning anything, then the application part is very, very important. So please look for the on the basis of your application. So can anybody also, here it is a very good question that which kind of samples that we can do by this phase contrast microscopy. So can anybody give some view on this? Any you can guess. Don't worry, if it is wrong, then it's fine. We are here to discuss, not to, giving some points on the basis of your right or wrong answers. So you are feel free to, guess, or it's fine. Okay, so I think nobody wants to guess. So let's see that what could be. Okay, so here. Okay, so as I have said about the phase contrast microscopy, few slides back, I was saying that I will be, discussing about in a little more detail. So here, this is the phase contrast imaging of transparent thin specimen. Okay, so this is a cell, you can say. Okay, yeah, this is a cell. So this is every cell is having a nucleus. Okay, and this is the surrounding of that cell. Okay, this is the adherent cell, and this is the nucleus, and this is the cell H. So when the light comes or the light falls, so what exactly happens? The this the nucleus is having a different refractive index as compared to the surrounding. Okay, so very small difference exists between the refractive indices of the cells and their surroundings aqueous solution. Okay, and within the cells between the cytoplasm and the cell nucleus. So here, as I said, that due to the change in the refractive index, the phase the change in phase will occur. So no significant refractive index difference occur in the cover slip and the surrounding medium. But here when we will use the biological sample, so here the refractive index will be the change. So the phase contrast makes this tiny difference. Okay, so there will be the difference in the refractive indices visible by the use of optical manipulation. For example, it translates them into difference in intensity that can be visually observed and the recorded. Okay, so here due to the change in the refractive index, the phase there you can see there is a delta 1 and the delta 2. So here since the light will pass here, firstly the light phase will be like this, and then the phase change will occur because here it is passing through the cell edge, but again when it is passing through the nucleus of the cells, then the phase change will be the different. Okay, so here due to the change in the refractive index, change in phase will occur. So here you can also see the examples of the this is the cells. Okay, so when you will be, this is the actual real image that I have taken from the slides of the lecture. So this is the living cells in the two kind of microscopy. This is the bright field and this is the phase contrast. So here you can clearly see now. So few minutes ago I was saying that please look behind the application also. So here if you want to see your cells, or if you are having some biological samples and you do not have any idea about which kind of microscopy you have to opt. So if you will go for the bright field, then you won't be able to get a good image. Okay, and if you will choose a phase contrast microscopy, then you can clearly see that how clearly you can see. Okay, this is the surrounding, this is the nucleus. So this is the main application. So here we will be definitely going for the phase contrast. So here the answer will be the thin, transparent and the unstained biological samples. So if you are having even a thin, so here you can see the phase image of the transparent thin specimen. So phase contrast microscopy can be used for the, here also you don't need to stain any your cells or the tissues. Please guys be on time. So sorry for the interruption. Okay. So So, okay, so this is the one kind of a, I when I was going through the lectures that I found it is also very good. So the hollow formation surrounding the feature appears in which kind of you can say the microscopy. There is a DIC microscopy, polarized microscopy, phase contrast microscopy, fluorescence microscopy. So if anybody remembered while going through the lectures that where you have seen the halo formation, can anybody answer? If you, okay, fine. Anybody has seen any other in any other microscopy? Guys, please speak up. Please say something. There are so many peoples. Same. Okay. Yeah, this is also fine, but at least say something. Same. Okay, fine. Okay, so let's see that the people are guessing the phase contrast microscopy. Okay, so I think you guys have gone through the lectures because you have guessed the right thing. So this is a kind of a image. Okay, so here you can see you can see there is a ring kind of a shape you can find. Okay, so in the phase contrast microscopy there is um, this is basically used to see the living cells in the culture media, but typically results in halos surrounding the outlines of the edge feature. So this is your it is the halos. So the halos arises because of the configurational parameters of the phase contrast microscope optical system. Okay, so here also this is the circular phase retarding ring located in the objective phase plate also transmit a small degree of diffracted light from the specimen. So this is basically, so when you will see the optical arrangement in the phase contrast microscopy. Okay, I think I have not included. Okay, maybe in the further slides I can show if I will be incorporated that. So here there will be the phase retarding ring located in the objective phase plate. Okay, so when the light will transmit, then there will be the some diffracted light will also, there will be some small degree of the diffracted light from the specimen, so that light will also pass. Okay, so for an example, if you are having a aperture of any particular dimension, but there will be also some light is coming from the side or from somewhere else. Okay, so suppose if you are having a torch. Okay, let's give me a very easy example. So if you will having a torch. Okay, so in the torch there will be a high intensity at the middle, but at the surroundings you will have some kind of a shadow kind of a structure. Okay, so here also when the light is passing through the aperture or the path, there will be some small degree of the diffracted light from the specimen will also comes. Okay, so that will gives this halo formation. So the presence and the severity of the halos in phase contrast is depend on the refractive index difference between the specimen and the surrounding medium. So this is also because of the same reason, because there will be the refractive index difference between the specimen and the surrounding medium. So when the light or any comes from the lamp or any light sources, then there will be the diffracted light from the specimen will come, and that will lead to give this halo formation because of the difference in the refractive index of the surrounding medium and the specimen. Okay, so there is one more very separated lecture. Differential interface contrast microscopy is used for thin specimen with reflected mode, thick specimen with reflected mode, thin specimen with transmission mode, thick specimen with transmission mode. So please give answer for this, and we'll see. Okay, so I will do one thing, because I will I don't know that how many students have gone through the lectures. So because there are very few who are speaking and giving the answers. So I will leave this question for you guys, and I will ask this question to those who are not giving answer in the next class. So this one question will be, I will ask in the next class, that please note that in the DIC microscope is used for what? It is again kind of a application, you can say, for the thin specimen with reflected, thick specimen with reflected, and there is in transmission and the, yeah, in transmission mode. So please answer this later. Okay, so have you in the lectures there is a prism. Okay, so when you see the optical arrangement of any of the microscopy, if you have remembered, then there is one prism. So this I will give you a hint that this prism is twice. I mean, in this optical arrangement, this has been used twice. So the Wollaston prism is made by two prisms fused each other having similar refractive index, different refractive index, similar reflectivity, different reflectivity. So if anybody is knowing that about the Wollaston prism, then they can answer this question very easily. Can anybody guess or answer? Nobody. Okay, fine. Okay, let's see. Okay, yeah. So in the differential interface contrast microscopy, so you will be having a Wollaston prism. So the right answer is correct, that there is the Wollaston prism are a type of prism made of two layer of a crystalline substance such as quartz, which due to the variation, or you can say the one who has said the difference of the refractive index depending on the polarization of light, splits the light according to its polarization. So the right answer is fine, but I will also here you can see in when you will be in the lecture, this is your Wollaston prism, and this is the also. So when the light is coming, this is the unpolarized light, so there will be the polarizing filter, and when filter at 45 degree prepares the light for the first Wollaston prism, and this is your second Wollaston prism. So this is the 45 degree polarized light will go to this first Wollaston prism, and this will separate the incoming light into the perpendicular polarized component. Okay, so again there will be the condenser lens. Okay, so this light will come, and this condenser lens will again accumulate this light to the specimen, and from the samples again this light will come and it will interact with the objective lens. So here this is splitting the polar 45 degree polarized light, but here when it is interact with the second Wollaston prism, so that will recombine the incoming perpendicular polarized light into the same polarization. So here this is recombining and this is the splitting. So here the polarized filter, it will give the 135-degree polarized light. Okay, so you can see that how in the lectures, because it is just asking about the Wollaston prism, so I'm just, it is also, you can just Google search about the Wollaston prism, that what how it looks here. Like when I was going through the lectures, I did some quick Wikipedia search. So here also you can having some guess or the, yeah, but this is having a very good what to say, this the lecture, in this lecture you can very well understand the exactly and how this phenomena works, or what is the working principle of the DIC. So the next question is, the which mode of optical microscopy gives the depth perception? Okay, so again I will again bring you in the same route. If you are having some samples which is having a depth, or which is having some thickness, then which kind of microscopy you will go? Again, this is the good question, that what is the microscopy that you would like to opt. So can anybody answer this very quickly? Okay, fine. Okay, so here you can see this is the image formation in the DIC mode. So here you are having some, suppose if you are having some samples which are having a thickness. Okay, so okay, let me say. So actually the answer is the DIC, not the polarized microscopy. So here you can again I will say that when I will generate. Okay, so when the light is coming from this light source, and when it is coming, so here image is generated from two identical bright field images being overlaid slightly offset from each other. So here it is not exactly, this is giving a shadowing image. Okay, the subsequent interference due to the phase difference converting changes in phase. So here this is actually giving you, it looks like it is giving a depth, but it is a kind of a shadow which is occurring because of the change or the difference in the refractive index. So suppose here the light is coming, okay, this is the specimen and the light is coming. So here two image will be the generated, but there is a difference in the phase. Okay, so when you overlap these two image on top of each other, then that will come as a shadow and that will give you a depth perception. So this is prisma orientation is different. So here the DIC image has appearance of a three-dimensional object under very oblique illumination, causing strong light and dark shadows on the corresponding. So here it is also exactly not exactly you can say image, whatever you are having a specimen, it is not given, it is the interference or yeah, it is also because of the interference of the kind of a constructive and the destructive, giving rise to the characteristic appearance in the three dimension. So when the light is coming, there will be the interference will also occur, and that will be giving a change in phase. And this is a kind of a shadow kind of a image will occur, so that will be giving a depth perception. It is not, that's why it is having a depth perception. The main you can, if you will be gone through the lecture, then you can easily understand this. So in the DIC mode you can easily find the depth perception. So cross polarized configuration is shown in which microscopy? So cross polarized, okay, so there is a polarized microscopy, DIC phase contrast, and the DIC and polarized. So cross polarized is also the same reason behind this is for the generating of the polarized light. So here this I have shown a few slides ago. So cross, this is cross polarized configuration. Okay, so cross polarized matlab here you are having a polarizer. Okay, this is a polarizer. So when the light is coming and it will pass through the polarizer, and cross polarizer means the analyzer is also one kind of a polarizer. So here you can see the direction, this is a horizontal and this is a perpendicular. So when these two directions are perpendicular to each other, then you can see the cross polarized. So in this configuration, the polarizer and the analyzer are crossed with no light passing through the optical system and a dark background present in the eyepiece. So here the cross or you can see the cross polarized configuration. So here you can see the cross polarized means the polarizer and the analyzer are having a perpendicular direction. And also in the requirement of the, so this is from the polarized microscopy, and this is the requirement in the DIC. We have also seen that we are using a polarizer. Okay, so which will generate, so as I have seen a few slides back, yeah, so here this is the polarizing filter, and this is also at the end. So here also we are using a polarizing filter, so in the DIC with to generate a polarized light. So the right answer will be the DIC and the polarized microscopy. Okay, so the polarized microscopy color image are formed due to the presence of what? So as I have shown some image, there is a polarizer, analyzer, lambda plate, polarizer, and the analyzer. So okay, so when you will be going through this slide, so in the polarized microscopy, so again here is you can see a very nice image of the, here you can guess the phases of your material. If you are having some material which are having a different kind of elements, then you can guess the phase of your material. So by using a lambda plate, you can guess the color images. Okay, or you can find the color image by using this lambda plate in the polarized microscopy. So here also the phase difference is the main reason behind this. So the part difference, okay, when this will, there is the analyzer, when the light will pass through this, there will be the part difference. Okay, so this part difference leads an extension of certain wavelengths in the light through the interference. So here also the interference will occur, so that because from the every part it is not always similar, all the light can pass. Okay, so there will be the path difference, and due to that certain colors remain from the white light, and that will create a beautiful colored picture in the polarized microscopy. Okay, so the main thing is the lambda plate. By using lambda plate we can get this color. So which optical microscope works efficiently in identifying multi-phase material? So just now I have showed you some multi-phase material. So here you can see this multi-phase, you can see a lot of phase. So here this is again the phase contrast microscopy. So this is also, sorry, okay, so this is a polarized microscopy, not phase contrast. So this was also from the polarized light microscopy. So here you can see this is yttrium, barium, copper oxide. So these are having different phases. So here you can see by using a polarized light microscopy, you can see the multi-phase material, and also this is depends on the crystal orientation. So different crystals will generate a different part difference. Okay, so that will lead to give you a difference in the color of your faces. So here you can identify your faces. So this is your tetragonal face and this is your orthorhombic face. So maybe in the, okay, so this is a very good question. In fluorescence microscope, excitation and emission signals are differentiated by which? Excitation filter, emission filter, dichroic mirror, eyepiece. So maybe you all have gone through the lectures. Maybe it is, I think, in the last lecture. So there will be a phosphorescence microscopy, fluorescence microscopy. So here I will show you. So it is asking about the excitation and emission signals are differentiated by what? So it is a fluorescence microscopy. Okay, so here when the light is coming from the light source, and this is your excitation filter. Okay, so by excitation filter it is going to the dichroic mirror. So through the dichroic mirror, so it is like that it is only allowing, so when by reflecting the light will go here, but when the specimen will emit some light, so this dichroic mirror will only allow the light which is coming from the emission. Okay, so here it is asking about the excitation and the emission signals are differentiated. So this dichroic mirror is a very, sorry, this dichroic mirror is having a very important role to uh, distinguish between the exciting signal and the emitted signal. So when the specimen is emitted from here, then this will only allow the signals which are emitted from the specimen, and only the signals will pass through this dichroic mirror. So here the option is this. Again, the time lag is shown by the fluorescence, phosphorescence, transmittance, reflectance. So this is very simple. This we have also know from our in many courses. This you have seen. Okay, so this is the fluorescence and this phenomena. Okay, so when we will excite, okay, so when we will give some illumination or the light source, okay, then the this from the exciting state in the fluorescence, this will go to the excited state, and immediately the electron will come to the ground state. Okay, so this will not taking any time, but in the case of phosphorescence, when you will excite from the ground state to the excited state, it will not directly come to the ground state. It will be your carrier will be going to the state which is at the between, which is called the triplet excited state, and after this it will come to the ground state. Okay, so this is a very quick phenomena. Okay, and this is a very slow phenomena. Okay, there is also an example where you can see the time scale. So from this ground state to the excited state, there will be some vibration losses, and it will come to the again the same state. So here the time scale is 10 to the power 10 to 10 to the power 7 per second, but this can be higher than this. Okay, so this is a slow process which is a phosphorescence, and the fluorescence is a very quick process, because here it is the carrier is coming by the mean of the state which is in between the ground state and the optical state. Okay, so the energy Difference between the observed and the emitted photon is bigger and the wavelength shift more pronounced. Okay, so here it is very simple that the phosphorescence will take a longer time than the fluorescence. So the answer is phosphorescence. So yeah, so because time, it is asking about the time lag or which is slow, you can say. So the phosphorescence, because the fluorescence, the process is very, very quick. So I think we are 8 now, and we have discussed some question. So okay, I will. So do you guys having any questions or any queries that you would like to ask? Because at the last I was just covering the questions because of the lack of time. Seven. Okay, lecture. So actually, we are having every week Tuesday in the same time, 7 to 8 p.m. So we will be discussing. Okay, so it is like that. This week we have discussed the week two, your kind of week two lectures, some problems related to the week two lectures. Again, in the next week, I will take some questions or some problems related to your week three lectures. So it will be going like this. So if you will have any doubt or any queries or any questions, then you can also ask. And if okay, okay, sure, sure, why not? Okay, okay, I will. So okay, let me do one thing. Uh, So in the chat box, you can see my email ID. So if you will have any query or any question that you would like to cover, then this is also one of the very good mode that you can ask by this by emailing me, and I would try to answer your question or any doubt in the next session. So that will also help you to learn better. Yeah, sure. Okay. Okay, okay. Okay, okay, fine, fine, sure, sure. Okay, okay, yeah, yeah. So, does anybody is having any more questions? Week two assignment. Okay, I do not have the questions. Can you ask? Can you? Okay, I will note down. What is the number? Okay, okay, I will check because I haven't. I was just preparing the questions. So, question number six. Okay, I will see and I will let you know because I do not have the questions right now. Okay, so does anybody also having any query or in this lecture? Yeah, yeah, actually, I'm also not sure, but okay, I will also ask to the other TAs because I do not have the answer. Yeah, yeah. Okay, okay, sure. I will ask the other TAs and will let you know. Huh. Yeah, yeah. Yeah. Okay, okay. Oh, okay, okay, fine. Yeah, 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 yeah. So actually, yeah, you are right. But the thing is like that only because in uh, the prophysis is also a kind of covering the only the part which is in the content of the course. So if they will start from the very very basics, then that might take a very long time. So please try from your side also to so that yeah yeah yeah. So there are a lot of lectures and but yeah, you have to put some extra effort who are not from the same background. So make sure for that. Yeah. Sh- yeah, sure. So if you do not have any questions, then you may log out. What you want? Yeah, actually that I do not have, so I have to check that in the portal because I haven't seen your assignments. I was just going through some related questions. So I will see the questions and I will try to solve that questions because I have to also see some lectures to see that. But anyway, I will see your question number 6, right? Okay, fine. Okay.